in the Department of Kinesiology and Health Sciences, and I'll be chairing this session. We have 4 excellent speakers, or well, at least 3, I'll just go over the order of presentations today. So the first speaker is Professor Robyn Murphy from La Trobe University. That'll be followed by my presentation, and then Professor Arthur Chang from York University will be speaking third, and the fourth speaker is Professor Nicholas Place from Lausanne University. Each presentation, it will be for 20 or 25 minutes, so we should have 5 or 10 minutes after each presentation for questions from the audience. So it's my pleasure to welcome and introduce our first speaker, Professor Robyn Murphy. Robyn is currently a professor in the Department of Biochemistry and Genetics at La Trobe University, and she'll be talking to us today about furthering insights into SR calcium ATPase, or SERCA, in skeletal muscle. Welcome.
Thanks, Russ. And there's always someone who apologises for the croaky voice, and it's me this year. I might have picked it up somewhere in the Rockies, which is where I managed to spend a couple of days with Barney Frankish, and I'm going to show some of his work in the talk today. So thanks for being here. So I'm going to start off by talking about the metabolic demands of skeletal muscle really briefly, but essentially the metabolic demands will increase as we exercise, and the energy demands increase, and that comes alongside a commensurate increase in ATP utilisation. Now, where that ATP is being used primarily will be up here at the surface membrane to maintain excitation-contraction coupling, and the movement of sodium and potassium is particularly important to the polarisation of the membrane. The SERCA pump, which is where the focus will be, and I'm going to talk about that a little bit more, and also down here at the myosin and actin contractile units, the ATP consumption will be in those areas. So if we just have a look at SERCA here, one of the reasons why this is the sarcoplasmic reticulum, if you couldn't quite make out my drawing, but this is the sarcoplasmic reticulum, and it stores a lot of calcium, and work that I've done with Graham Lamb over the years and Cedric Lamboli when we moved to human tissue showed us that we have about 1 millimolar total calcium inside the SR. So any increase in calcium outside in the cytoplasm is going to have a difficult time getting in unless there's a very strong buffer inside, and there is, and that's called calsequestrin. So calsequestrin binds 60 to 80 calcium molecules per unit, and so basically your driving force is one that's favourable to calcium being taken up into the SR. A lot of the work that I do really addresses muscle heterogeneity, and this is I really like this image of Vels actually, just showing that side by side in the same muscle sample you have type 1 in the blue and type 2A and type 2X muscle fibres. And as Zen was showing us earlier today, they're very different in metabolic and contractile properties. And what we all know is that muscle is very plastic, and so in response to an intervention or given an age or a genetic state, you can alter your fibre type distribution. And this is just showing some data here. Most of these have been shown through single muscle fibre Western blotting and probing with the myosin heavy chain 2 or 1 antibody. And this we actually just published recently showing that there's a difference in the fibre type distribution between a type 2 diabetic and a control individual. And this is looking at older people who underwent training, and there's a shift in fibre type, and then younger people. So this is not new, but it's just to remind us that muscle is very plastic. And if that is the case, then it's particularly important to look at things in individual muscle fibres. And indeed, that's what this study from, that's been done before. This study showed. So Zentesi and Fink looked at the ATPase rates in the different fibre types that they isolated from human muscle biopsies. So you can see the numbers are quite small when you start to have any with 2X in them. And basally there's a moderate consumption of ATP. It is different as you go up to the 2X, but this is the most important one, and that is the maximal total ATPase consumption, which was under maximal isometric force. And of this maximal usage, the amount used by the sodium potassium ATPase remains fairly constant at about 10% of the total usage. the myosin ATPase goes up, and also the amount that the SERCA will use increases, and it increases to a greater extent in these last two ATPases in the type 2 muscle fibres. So we want to drill down and have a look at what these muscle fibres will be doing in terms of the SERCA and other regulatory factors. So as the title of this session refers to, calcium is involved in a lot, and that's for you, Lawrence, the involvement of calcium. And what I want to talk about is the movement of calcium around the cell. So calcium isn't uniform across the entire region of the myofibre. So that's what we're going to drill down to. And there's constant competition for calcium within the myofibre, but the key player is the SERCA pump because it has a high affinity for calcium. And not surprisingly, perhaps this is a regulated protein. What we tend to do in the lab a lot is use single muscle fibres. This can be either a freshly obtained muscle fibre that's dissected under paraffin oil in a dish, and there's a single fibre segment that's being collected there. The length of these are 3 to 5 millimetres. The diameter of human fibres are in the order of 50 to 60 microns in a human vastus lateralis. And the amount of protein that you get is about 4 micrograms of total protein. But much more favourable in many circumstances is to use freeze-dried tissue where you can't get quite the same length of muscle fibre, but you're able to use that muscle, years into after having obtained it. So either of these methods are what we use when we want to have a look at proteins in isolated muscle fibres, and then we can address muscle heterogeneity by doing that. So this is what the a Western would look like. These are individual fibres that have been loaded, so they are different in size because the amount of protein in a muscle fibre is most dependent on the diameter, not the length, and that's a very hard measure to make under the microscope, and we don't try to do that because we'll use the stain-free, sorry, I didn't mean to do that straight away, the stain-free gel to tell us what the total protein is. So you can see that these individual fibres were predominantly containing heavy chain 2 or 1. And I just wanted to show this because I think it's particularly important to think about loading controls. And if you're going to look at muscle heterogeneity or something that will influence the fibre type distribution, if you use a protein such as GAPDH, it's not ideal because GAPDH, as an example, is twofold higher in type 2 muscle fibres compared to type 1 muscle fibres, whether the samples are from a young person or from an older person. And also the amount of GAPDH in an old muscle from an older person is decreased in type 1 and in type 2 muscle fibres. So that as a loading control is really not a great one. And we will use the myosin heavy chain, as you see across here, when we're doing them. So this is the when we load a muscle fibre on a gel, and you can see that this one has myosin heavy chain 2. It has the isoform SERCA1, and it has the calsequestrin isoform calsequestrin 1. And conversely, there's a fibre here that's a slow twitch muscle fibre having the SERCA2A and the calsequestrin 2 protein. And this is, next to it is a hybrid fibre. So that's sort of showing a bit of the methodology that we can use to break the muscle down into the different fibre types. So the other thing that we had done with Graham in rat muscle was to show there was around about 1 millimolar calcium in the fibres. Now this is actually fairly similar in humans, type 1 and type 2 fibres. This is the endogenous content of calcium in the SR. And it is significantly different, but not hugely. But you can see that the maximal capacity goes up quite a lot, so almost twofold. And there is a difference. The type 2 muscle fibres, or the SERCA1 containing SR, can take more calcium into them. So what we did with Barney was to say, well, that's type 1 and type 2A, but of course there's an array of fibre types there, and 2X are the ones that we don't know much about, and often we won't include data for them because we just don't get enough 2X fibre types. But here what Barney did was once again load all the single fibres on, and he looked at the SERCA1 that was present in each of the samples, and these are in the type 1, the 1-2 hybrids, the 2A, 2AX and 2X fibre types, and you can see that there's more SERCA1 in those particular fibre types. And that's all well and good, but what I want to draw our attention to now is that If we look at the 2A, 2AX and the 2X, there isn't a great deal of difference in the abundance of SERCA. But up here, you've got a threefold difference in the ATPase consumption of those muscle fibres. So the SERCA itself can't be playing the role on its own, and this is where the regulation comes into it. How is that the case? Well, that takes us to a couple of regulatory proteins, phospholamban and sarcolipin. And these particular proteins can bind. They bind to the same binding site on SERCA, so they can't bind at the same time, but they're present. And phospholamban was first identified in cardiac muscle, and it inhibits the uptake of calcium into the SR if it binds to the SERCA. And it's got a higher affinity for SERCA1 than SERCA2A, so you'd expect there to be more in type 1 fibres if that was the case. And it also comes in a monomeric form and a pentameric form. And it's the monomeric form, which is about 6 kilodaltons, that is the most inhibitory form. And then it can be phosphorylated. I think there'll be some of that presented throughout this symposium. And when you have phospholamban phosphorylated interacting with the SERCA, then you will change the relaxation rate, and that's been shown in the transgenic mice. So this once again is from Val's work where Russ and Val were interested in looking at phospholamban in the single muscle fibres, and you can see that these very light ones coincide with the red fibres over here being the type 2X, so it looks like there's very little phospholamban in the type 2X muscle fibres. So we went in and used our semi-quantitative Western blot approach, which allows you to calibrate the system so you can see where the bottom of detection is, and so you can identify these. And to do that, as I said, type 2X fibres are hard to come by, but they're not if you take a particular cohort. And here what we used was individuals who had non-metastatic prostate cancer, 6 people in total, and they had a lot of type 2X muscle fibres. So mindful of the data here, we had endurance-trained individuals and healthy untrained, and these individuals. But what we wanted to know was not the fibre types within the individuals, but across the spectrum, so that we could start to understand what 2X fibres were looking like. And here are the data that Barney's produced. The phospholamban is a 6 kilodalton protein, and sarcolipin, I'm going to show you, is 4 kilodalton, so it's quite challenging to get these proteins. But what we can see, whether it's pentameric or monomeric phospholamban, there's much less in the fibres that contain 2X, which coincides with what Val and Russ had shown. So sarcolipin's been proposed to act as a counterpart to phospholamban in the fast twitch muscle. So if that's the case, it may regulate SERCA1 through a different process, and certainly it's been tied into an enhanced maximal SERCA activity, and very much tied in with thermogenesis and the regulation of heat generation by muscles in endotherms such as ourselves. So when Barney looked at sarcolipin, indeed we found that there was more sarcolipin in the 2A, 2AX and 2X muscle fibre types. So this regulation is possible because of these proteins that compete together. So the thing that we found out was that whilst phospholamban is shown here, there is still a little bit of sarcolipin there, and you can have phospholamban and sarcolipin present in the same muscle fibres, and that wasn't really understood until we broke down to these single muscle fibres. So that's those proteins, but there are a couple of other proteins that might regulate SERCA in similar ways. They've been less explored, and we haven't explored them. And I read less about these ones, but what I wanted to say is that there could be an array of proteins that come in and regulate SERCA in order to slow down the function of SERCA, and you'll see some data from Russ, I believe, showing that SERCA function is dampened, and how's that happening? So one of the things that SERCA will do, and something that's sort of held back our understanding of SERCA function, has been around being able to measure the function of SERCA in an individual muscle fibre. I've collaborated with Brad Lornakonis over the last 12 years, and this is a photo of Aldo, his PhD student who's just completed. And what Aldo's been able to do is show that the heat production in an individual muscle fibre, and that heat production is coming from SERCA pump activity. So we now have this tool that's going to allow us to put sarcolipin or put phospholamban into the system and understand how it's regulating the ATPase function. So here is it's the same muscle fibre, and this is capsaicin. Capsaicin is an inhibitor of the SERCA activity. And when you see yellow, that is heat being produced. And when the capsaicin is added, over time you get to about 10 minutes, and you can see there's a lot less heat being produced. So this is your normal endothermic heat generation that you'll see. You can block that off, and then you can wash out the capsaicin and you start to get the heat generation again. So this is a tool, it's been published last year as a technique, and it's a beautiful technique that's going to allow us to look at individual muscle fibres at the level of ATPase function. So I want to talk now about something it'll be prevalent in this session, and that's about ryanodine receptor fragmentation. And this is from Nicholas's paper from a few years ago, showing that with exercise, there's a reduction in the full-length ryanodine receptor, and that's the 550-odd kilodalton protein just sitting here. I've just taken a snapshot of the graph showing the reduction in the full length, not the appearance of these other ones just here. And that's been of interest to a lot of people, including collaborators in Calgary, Tom and Martin, and that's where Barney's working now. And what we published just recently, it's in press, is that it is following some 6 by 30 second all-out bouts of exercise. So the difference here is very nicely, there's two aspects. One is there's whole muscle being looked at 3, 6 and 24 hours after exercise, because most of the work had been at 24 hours, but at what point is that fragmentation actually happening? So that was one of the questions that wanted to be addressed. And then also, what about the type 1 and the type 2 muscle fibres? So you can see here that there's type 1 here and type 2A, and these are other calcium proteins that I won't go through, but the important one is the ryanodine receptor. So it operates as a tetramer, but we see it as a monomer on the gels. And what you can see, similarly to the PLAS paper, was that at 24 hours you do get this reduction in the full-length ryanodine receptor, and you can see that this is happening already at 3 hours post the exercise. And over here in the single muscle fibres, you don't see the fragmentation, but you do see the decrease in the full length, and that's what we focused on in this particular part of the study. So you can see in this individual at 6 hours, in both type 1 and type 2 fibres, there seemed to be a reduction. When the data were crunched, in whole muscle, there's a reduction in the full-length ryanodine receptor at the lowest point, so whether it be 3, 6 or 24 hours. In type 1 fibres, it was variable and it didn't have any statistical difference, but in type 2 fibres, there was a statistical reduction in the ryanodine receptor full length. So if this is the case, then what's that actually doing inside the muscle? And one of the items explored in the paper was to say, well, if you have more calcium, that will trigger PGC1 alpha transcription, because the protein is not possible to be seen, in our hands at least, with decent antibodies. So you can see that the mRNA has gone up for PGC1 alpha by 3 hours, and then it comes down again. This is putting the PGC1 and the ryanodine receptor on the same graph. And whilst it looks like they might be correlated, there isn't actually a significant correlation there. But certainly this leak of calcium... when the ryanodine receptor fragments, is there this leak of calcium that will come out of the ryanodine receptor that we don't really want? And why is the calcium coming out? So it could be to drive some mitochondrial biogenesis, and that's one thing that's being explored. But something else that we're doing with Brad, and this is a photo of all of Brad's lab, because they all sort of contribute to this, that is to start to have a look at the calcium in its compartments inside the same muscle fibre. It's such elegant biophysical work. And here you can see that most of the calcium is sitting in the SR. The concentration is quite similar to what we've reported, but you can also see that there's a small amount that's in the mitochondria, and there's some that's sitting in the T-tubule, and there's also some that's unknown its location, so we don't quite know that. If we look at the type 1 and the type 2, the numbers sort of look quite similar, but if I show them as a whole when we've got an intervention, they're similar, but they're different still. So this is where the the interest lies. So this is actually the amount of calcium and where the calcium is in absolute amounts, so not just a relative amount, it's absolute amounts in human muscle fibres before this exercise, and then similarly high intensity training, interval exercise, 24 hours later, and there's a shift of calcium from in the type 1 fibres to be double in the mitochondria and 5-fold shift in the type 2 fibres. So there's something happening that's allowing the calcium to go into the mitochondria. And why would it be going into the mitochondria? It was quite contentious 30 years ago where, I forget the name of the person now, but where they did EM and basically concluded that there was no calcium in the mitochondria. 10 years ago, I looked at it using a less sensitive method and I couldn't find calcium in the mitochondria. So being able to see this and being able to quantify it really does allow us to say there is calcium and how much there is. It's in the order of 0.06 millimolar, 0.05 millimolar, so it's not nearly as much as in the SR. It's only a small amount, but it goes up significantly. So we're trying to explore what that might mean. And one of the ways that we are exploring this is to have a look at the compartmentalisation of the calcium in animals. So all the work I've shown to date was in humans, but now we're going to go to the animals and have a look at animals that are young and older, so 3 months old and 24 months old. And with the older animals, have a look after they've been given access to a running wheel for 35 days. And as usual, they run quite a bit. And then compare the localisation of the calcium in the old mice and before and after exercise. So when we look at the total calcium in those animals, it's very similar between young and old, whether they've been given a running wheel or not. So that's not surprising to us who sort of think a lot about calcium. But when we have a look at the SR calcium content, there is a difference, what's heading to be a difference. And where you have less calcium in the SR in an older individual, and it looks like there's a bit of a recovery. The numbers are not strong enough yet to be able to say that there's a significant difference there. But there does seem to be a redistribution into the mitochondria. So here you've got the amount in the mitochondria in the order of this 0.02 or 3 millimolars in the young animals, whether they've run or not. And in the older animals, there's a lot more. It's in the order of 0.23 millimoles, so it's quite high. And then that reduces after there's a running session. So I think what we would say as people interested in exercise and the benefits of exercise, we'd be seeing this as this is a good thing to have it coming down, and this was not such a good thing. But we don't have the evidence to say that what this is doing and what it's driving. It will drive oxphos, it'll drive biogenesis, but what else is it driving? And I think the Mito people here might be able to share some aspects of that. So this is just a summary there. There's more in the mitochondria, but I really want to make a note that this is in absolute terms and it's very small. So we've got to be mindful of that. So altogether, that where there's a loss of ryanodine receptor integrity, there seems to be a shift in the calcium movements within a muscle fibre. And it prevents, when it's possible, a chronic increase in calcium that might go on and trigger calcium-dependent events such as the activation of calpains or some kinases that you don't necessarily want to be activated. The SR is very good at taking calcium up, and Russ and I had a brief chat before this saying, well, why does it stop being as good as it really is all the time? So this is the void of what we don't quite know. The redistribution of the cellular calcium, there's less in the SR, more in the cytoplasm and the mitochondria, albeit in smaller amounts. And as a final point, the sarcolipin and phospholamban are certainly key to regulating the uptake into the SR. And now with those beautiful tools that Brad's group have put together, we're going to start to explore that in greater detail. So with that, I've listed the various people, and you'll see that Barney's there in a couple of places because he did work at La Trobe and now he's doing work at Calgary, and Brad's lab and others involved. So thank you very much.
Thanks very much, Robyn. We have about 5 minutes for questions.
Wim Draven from Ghent University. Thanks for a nice presentation. The 6 times Wingate test, is it still physiological, or is that more of a pathophysiological process or induction?
I can hear Eric laughing about this. So it's a discussion that I haven't heard. I don't know. I'm open to hearing whether or not we should be using that. I guess in these types of tests, what you're trying to do is to push the system to say what might be happening. Is it physiological? Well, high intensity is. Is it different from a high intensity bout? I'll ask a question back to you.
Yeah, I think it's a little bit a physiological way of destroying the system, but I'm not sure if you'd agree with that, destroying the mitochondria in a physiological way, like you do muscle damage in eccentric exercise. I think it's a bit similar to that. If you
to do this, and all the work on this has been done in the high intensity part, not in the with an eccentric load. Mm-hmm. And whether that same shift happens with eccentric, I'm not sure, but given that the calcium goes up in the cytoplasm, I'd suggest that it would be, we would see a similar response, because this is not about the exercise, this is about the calcium going up in the cytoplasm, and where does it go, because it can't stay up. And what I do know, and you know this too, is with the calpain and calpain 3, it's activated with eccentric. It's not activated with this type of exercise. And that's where you've got some damage and some, you've got to go and repair the muscle. So it is perhaps that's what we need to have a look at as well, to see whether or not that process is happening in there. I appreciate that as guidance.
Okay, thank you.
Thanks, Vim.
Thanks, Robyn, for a great talk, very comprehensive. I'm really interested in the local heat production measurement, but also in the implications of that. Do you have any thoughts or evidence that local heat production compromises SERCA function and that it would alter calcium compartmentalisation or contribute to fatigue?
That's a good question. I don't have any evidence that it alters it, and these experiments are not done at 37 degrees, and so that is something to always be a bit mindful of, I guess, to say, well, if it's at 37, then what? What we do know is that when Brad has looked at mouse fibres and looked at toad fibres, you don't get the heat production in a toad fibre under the same conditions. And that's an ectotherm. It doesn't need heat generation for homeostasis. And so using that toad is a good sort of control to say it's not happening there. So I don't think that there's going to be an interplay that's problematic there, but that's the answer.
I wonder if just hitting it with capsaicin and then contracting it might reveal something.
The capsaicin?
Like generate some heat, compromise the system, and then measure compartmentalization or contractility, and understand if SERCA is functioning properly after heat stress, local heat stress.
Yeah, the capsaicin is not the tool we're using to actually. That was a tool to say, can we measure this? Yes. What we want to do is use things that are in the cell, like sarcolipin and phospholamban, and say recapitulate it. But so at the moment we haven't made those proteins to be able to do it, and they're a little bit sticky to make. So there's a bit of a hold up on being able to do that.
There, thanks.
Thanks.
And we have time for one more question.
Thank you. Robyn, very nice work. I really like your single fiber approach. I was wondering about sarcolipin, because I don't think we know so much about it. Phospholamban is phosphorylated during exercise, and that affects its function. But sarcolipin do you know, is it phosphorylated? Is it regulated anyway during exercise?
I don't think it is. Is it, Russ?
There is evidence that threonine 5 in sarcolipin can be phosphorylated by CAM kinase. Whether it's phosphorylated during exercise, I don't think there's any evidence for that.
Well, CAM kinase is probably activated, right? So you would think
just
so.
Hasn't been measured. Yeah.
Does it affect the function? Do you know?
It does, similar to phospholamban. Yeah. Okay.
Well, thank you.
Thanks, Eric.
I noticed you had an antibody against it, because I know it's been quite difficult to
sarcolipin.
find.
Yeah, Barney's the expert.
Okay.
Oh, We got it from these guys. So Russ and Val's the expert. Barney was in my lab the expert at running the single fibre gels. Yeah, but yeah, it's very tricky because it's very small. It runs off. I don't think it's the antibody as much as the conditions that you need to actually keep the protein on the gel.
Okay, thank you.
Thank you. I think we'll have to do one quick question, or?
That's one quick.
Okay.
Usually, increasing calcium is a danger signal for me inside the cell as well as in the mitochondria. Did you correlate it to the death of mitochondria, or did you check it, for example, in persons, in people with decreased mitochondrial function, such in diabetes?
Yeah, so the increases in this calcium is not enough to trigger contraction, and it's not enough to even trigger some of the calcium-dependent events. So it's quite a small increase, and we haven't looked at the mitochondrial death or cell death, but that's not happening. So it sits below that sort of extreme level of calcium. And one of the things that I do is measure calpain activation to say where is the calcium and its concentration sitting. Thank you.